Hello, everyone. Thank you for joining us for this webinar series on precision imaging agents and theranostics concept to clinic. My name is Sridhar Nimagada. I'm a professor in radiology at Johns Hopkins University School of Medicine. In this talk, I will focus on a class of drugs called antibodies. Antibodies are large molecules. They are approximately 150 kilodalton in size. They exhibit high specificity to their target. They also exhibit very unique in vivo behavior because of their large size. I will discuss how imaging methods we are developing can be used to understand antibody activity at the tumor. We hope that the type of measurements we make will enable us to optimize therapeutic regimens to improve efficacy and also in drug development. Before we go into detail about antibody therapeutics and their applications in cancer, let's take a look at what factors broadly define the success of anti-cancer therapy. We know very well that the tumor heterogeneity is a major factor in determining tumor's response to therapy. Success of molecularly targeted therapeutics that are targeting either HER2, EGFR, or BRAF are some of the examples. We also know that host heterogeneity is another major factor that contributes to therapy success or failure. Immune fitness and microbiome heterogeneity are examples of how host heterogeneity contributes to therapy success. Third one is drug itself. Some of the factors that define success of a therapeutic or a drug are its affinity for the target, specificity and selectivity for the target, ADME properties of the drug, and the resulting biological activity. These data are commonly obtained using measurements made either in vitro or ex vivo using blood or tissue samples. For most of the drugs, biologic activity depends upon how drug interacts with the target on the tumor cells. Therefore, blood measurements do not always accurately reflect activity of the drug at the tumor. Particularly in the case of solid tumors, our knowledge of drug exposure and concentration at the tumor is very limited. We are making strides in this aspect for small molecules and peptides, but much to be learned about antibodies and other macromolecular therapeutics. Also, small molecules have better permeability into tumors than macromolecular drugs, such as antibodies. So, in this lecture, I will focus on addressing the challenges posed by antibody therapeutics and their measurements of activity at the tumor. In the last couple of decades, antibodies have emerged as a highly successful class of therapeutics, with approval rates nearly twice that of low molecular weight agents. Some of the factors contributing to that success are high specificity and selectivity of antibodies that results in few off-target effects. For some antibody therapeutics, maximum tolerated dose is rarely reached. Therefore, one could give high enough concentration of the drug to achieve the desired occupancy at the tumor or the saturation effect at the tumor to observe desired therapeutic effect. Because of these advantages, the antibody therapeutic development has seen significant growth with more than 500 antibodies in various phases of clinical testing. Pharmacokinetics of antibodies, similar to low molecular weight agents, are measured in the blood or using collected cells and tissues by means of assays such as mass spectrometry or thermal shift assay, etc. However, none of those approaches provide a quantitative picture of drug activity in the tumor in real time. Moreover, advances in antibody engineering have led to the development of a variety of antibody formats, including naked antibodies, antibody drug conjugates, bispecific antibodies, and paratopic antibodies, to name a few. Therefore, we need new tools to understand the pharmacology of these new antibody formats at the tumor, particularly non-invasive tools to make such measurements in vivo and in real time without the need for collecting tissue specimens. One way to quantify antibody biodistribution in vivo is by using a radiolabeled analog of an antibody of interest that allows for non-invasive quantification of the target expression that antibody binds to, as well as provides insights into the pharmacokinetics of that antibody in different tissues and tumors. Here, we are seeing an example of zirconium-89 radiolabeled edzolizumab, a PD-L1 antibody, and provides a quantitative picture of tissue response kinetics, including in the tumor. That radioactivity accumulation in tumors can also be used to correlate with response or clinical responses, as you see on the right. However, keep in mind that tumor accumulation of antibody is a combination of target expression and drug exposure, therefore is a poor reflection of the serum concentrations of the antibodies. The antibodies have many advantages, as we have learned in the previous slides, such as high selectivity, few off-target effects, and long half-life. But they also have few disadvantages. For example, antibodies suffer from poor tumor penetration due to their large size, which is approximately 150 kilodaltons. This is because the nature of the solid tumors that present active and passive barriers to therapeutics. Poor vasculature and high interstitial pressure are some of the factors that keep therapeutics out of the tumors, and those effects are prominent in the case of antibodies. Antibodies are also susceptible to a phenomenon called the target-mediated drug disposition. Target-bound antibodies could be internalized and metabolized, therefore reducing tumor concentrations of the antibodies. The higher the density and internalization rate of the target, the larger the effect these processes have on tumor and circulating antibody concentrations. Also, development of anti-therapeutic antibodies is another factor that could influence antibody concentrations in the serum as well as in the tumor. In essence, antibody concentrations in the blood do not really reflect the concentrations in the tumors. This has been a missing piece in establishing the relevance of serum drug concentrations to that in the tumor in order to correlate with therapy response. To characterize in vivo distribution and pharmacology of antibody therapeutics, currently we use a drug-centric approach, wherein we take the generated radiolabeled analog of a therapeutic agent and study its pharmacokinetics and in vivo distribution. If we have different antibodies that bind the same target, the corresponding imaging agents could give a slightly different picture of target expression and drug distribution. Why? Those antibodies, even though they bind the same target, or whether it's a therapeutic or an imaging agent, are distinct from each other and therefore are subjected to different tumor intrinsic and extrinsic factors, as well as biophysical properties unique to that antibody. While these type of studies provide very valuable information about target expression and drug distribution, they're also very drug-centric. Also, radiolabeled antibodies take several days to provide good contrast images, and thus making it difficult to fit these studies within the routine clinical workflow. Furthermore, we are seeing significant changes in antibody development and approvals. For example, multiple antibodies are approved that bind to the same target, and the same antibody is approved for multiple indications. Often, and the efficacy of those antibody therapeutics is often assessed using either overall survival or progression-free survival that a long time, with no near-term pharmacodynamic measures used, specifically those that mediate the tumor. Therefore, generating a radiolabeled version of an antibody, even though it provides very valuable information, and to study them for in vivo pharmacology is resource intensive and technically challenging, even in a preclinical setting, and more so in clinical trials. We want to change the focus from the drug-centric approach to a target-centric approach. In this approach, as shown in the cartoon here, one could get a picture of the baseline target expression levels and could also monitor the changes in target levels and infer the activity of the antibody therapeutic in the tumors while the patient is undergoing treatment, irrespective of the antibody therapeutics we are using. To quantify the changes in target levels, we propose to take advantage of low molecular weight agents, which often have better permeability and faster kinetics than antibodies. and therefore would allow us to quantify the changes in target kinetics quickly and in a time frame that fits within the clinical workflow. Compared to examples we have learned from low molecular weight agents, the difference here is that we are monitoring the pharmacologic activity of a macromolecule using a low molecular weight imaging agent. However, few conditions need to be met for this approach to work. For this approach to work, small molecule derived imaging agents we are choosing need to have faster pharmacokinetics and low affinity than that of the antibody. As an analogy, antibody is similar to a sumo wrestler. It's big and slow to move about within the tumor interstitium. Small molecule-based imaging agents, on the other hand, exhibit faster pharmacokinetics and can only bind accessible target or protein expression sites. Also, they cannot displace target-bound antibody at the concentrations we are using for an imaging agent. Therefore, accessible targets that are measured using a low molecular weight PET imaging agent gives information on the target or protein of interest. These measurements are antibody independent and account for all intrinsic parameters imposed by the tumor and extrinsic parameters related to antibody properties and kinetics. To test this approach, we chose programmed Death Ligand 1 as an example. PDL1 is an important target for guiding immune checkpoint therapies. Also, 3 anti-PD-11 antibodies have received approval and provide us a really nice model system to prove the concept. So now a brief introduction about programmed Death Ligand 1 or PDL1. PDL1 is an immune checkpoint protein that is expressed by a variety of cancers as an immune evasion mechanism. Clinical data suggests that those tumors with high PDL1 expression respond well to anti-PDL1 therapeutics, and also therapeutics targeting its receptor programmed Death1. Because of this importance, PDL1 detection by IHC is used as a biomarker to guide anti-PD1 and PDL1 therapeutics. Also, there has been a significant interest in the imaging community to develop PDL1-based imaging agent, particularly for positron emission tomography, to characterize PDL1 levels in the whole body non-invasively, and also to monitor PDL1 changes during therapy by PET. Towards quantifying PDL1 levels non-invasively, we and many others have developed a variety of imaging agents, specifically for detection by positron emission tomography or PET. Our efforts were initially focused on antibodies and more recently on peptides. Why? Peptides have faster pharmacokinetics, and peptide-based imaging agents often provide high contrast PET images within a few hours of the radiotracer injection. What we have here on the top left panel is a 14 amino acid cyclic peptide called the DK222 that is labeled with fluorine 18 radionuclide. F18 has a half-life of 110 minutes and perhaps the most desired radionuclide for clinical applications because of its high positron yielding nature and also a reasonable half-life for routine production and shipment to short distances. DK222 binds PDL1 with high affinity and exhibits a dissociation constant of approximately 5 to 8 nanomolar, as you see on the top right panel. Also, 18F-DK222, radio-labeled by aluminum fluoride method, binds to cancer cells in a PDL1 specific manner. And that 18F-DK222 uptake correlates with PDL1 density in the cells, as you can see in the lower panel. What these data tell us is that DK222 binds PDL1 with high affinity, and radio-labeled or 18F-DK222 uptake in vitro correlates with PDL1 density on cancer cells. In this slide, we are showing the in vivo pharmacokinetics and distribution properties of 18F-DK222. In mice that are bearing MBMB231 breast tumors and that are known to be PDL1 positive, and injected with 18F-DK222. By 60 minutes, we see nice accumulation of radioactivity in the tumor. These are the subcutaneous tumors as shown by red circles. As the peptides are cleared or through renal clearance, you can see that high amount of radioactivity can also be seen in the kidneys. but very little radioactivity accumulation can be seen in other tissues, thus providing high contrast images within 60 minutes. They're also PD-L1 specific. How do we know that it is PD-L1 specific? When in the blocking dose mice, where we are injecting a high amount of non-radioactive peptide, the competition between radioactive peptide and non-radioactive peptide reduces the radioactive peptide accumulation in the tumors, thus showing that the peptide accumulation in the tumor is PD-L1 specific. Also, this peptide DK222 accumulation is very low in tumors that are known to be PD-L1 negative, such as SIM149. Collectively, what this data tells us is that 18F-DK22 provides high contrast PD-L1 specific images within 60 minutes that can be blocked with excessive peptide administration. And it exhibits tractable pharmacokinetics in vivo. So therefore, we took advantage of this high contrast of the imaging agent to study the pharmacokinetics of PD-L1 therapeutics, which I will show in the next few slides. To demonstrate the feasibility of our approach, or of our target-centric approach to quantify the activity of antibody therapeutics at the tumor, we first injected melanoma tumor-bearing mice with different doses of anti-PD-L1 antibody erzolizumab. 24 hours after treatment with the antibody, we injected 18F-DK222 and acquired imaging at 60 minutes, followed by biodistribution studies. In theory, if the antibody engages the target, in this case tumor PD-L1, we should see less 18F-DK222 uptake in the tumors. As you can see from the images here, 18F-DK222 uptake in tumors is high in saline controls in the left panel. In contrast, there is a significant reduction of radioactivity uptake in mice treated with 20 milligrams per kg saturating dose of erzolizumab, as shown in the rightmost panel in the PET images, suggesting that the most of the tumor PD-L1 is engaged or occupied by erzolizumab. However, when we use subsaturating concentrations of erzolizumab, for example, 0.3 milligrams per kg dose, there is an appreciable reduction of 18F-DK222 uptake in the tumors. But you could still see 18F-DK222 uptake in tumors, indicating that some PD-L1 is not occupied by the antibody and therefore accessible for the 18F-DK222 to be bound. This leads to increased basically accumulation and signal intensity in these mice. Collectively, this in vivo study demonstrates that the feasibility that one could quantify dose-dependent changes in accessible PD-L1 levels within tumors during treatment and for any antibody therapeutic. Importantly, we can do this without interrupting therapy and for any PD-L1 antibody. Next, we decided to test our approach to compare the biological activity of different PD-L1 antibodies. As I mentioned earlier during this talk, there are 3 PD-L1 antibodies approved, and they have different biophysical properties, as you can see from the table in this slide. To test if they differ, or if these 3 antibodies differ in pharmacological activity in the tumor in vivo, we treated melanoma tumor-bearing mice with 1 milligram per kg dose of the antibodies and quantified the accessible PD-L1 level at 24 and 96 hours using 18F-DK222 by PET imaging and biodistribution studies. What we have observed is that 18F-DK222 uptake is high in PBS-treated controls, as well as nivolumab, which is an anti-PD-1 antibody, control antibody. In mice treated with PD-L1 antibodies, such as erzolizumab, avelumab, or durvalumab, there is a significant reduction in 18F-DK222 uptake in tumors, indicating that there is a low accessible PD-L1 levels in these tumors once they are treated with antibody. At 24 hours, all the antibodies have similar effect on PD-L1. That is, almost all the PD-L1 within the tumors is bound by the antibody. In contrast, the effects are very different at 96 hours of treatment with the antibodies. We can observe more accessible PD-L1 levels in the case of erzolizumab and the avelumab treated mice. than in the case of durvalumab treatment. In the case of irzolizumab and nivolumab, we see increased accumulation of 18F-DK222. But whereas in durvalumab, the 18F-DK22 accumulation is similar both at 24 and 96 hours, suggesting that durvalumab somehow engages on the PD-L1 within the tumors for longer periods of time than irzolizumab and nivolumab, at least in the model system that we are testing. Collectively, these data demonstrate that different antibodies have different pharmacokinetics and pharmacodynamics in the tumors, and that one could use a low molecular weight agent such as 18F-DK222 to quantify the differential effects of these different antibodies in the tumors, in vivo and non-invasively. Next, we investigated if our novel approach can be used for antibody dose finding and dose optimization. We wanted to show how one could use pharmacodynamics measurements made in the tumor bed by PET imaging can be used for antibody drug development. The current process for dose finding of antibody therapeutics involves gathering in vivo pharmacokinetics and ADME information in preclinical models and building mathematical models that take into account of the target recycling rates, target expression levels, and antibody affinity to come up with a suitable dose. Those models are then further refined with the data from early phase clinical development and again in late stage clinical development to identify a suitable dose for approval by Food and Drug Administration. Those models rarely use disease site occupancy data that is acquired in real time. Well, there are very few tools to make target occupancy measurements in real time at the disease site, particularly for antibodies. Sometimes it takes several hundreds of patients to find the optimal dose for an antibody, suggesting that there is scope to refine our current models. Therefore, we decided to apply our imaging concept for dose finding of antibodies. To test our imaging approach for antibody dose finding, we used anti-PD-L1 antibody irzolizumab as an example. In this experiment, mice with the MDMB231 breast tumors were treated with different doses of PD-L1 antibody irzolizumab for 24 hours. Following that, target occupancy data were acquired 120 minutes after the radiotracer injection. What we have observed, as you can see from the PET images, is that tumor PD-L1 was barely engaged by irzolizumab at 0.19 milligrams per kg dose or any dose below that. And most of the tumor PD-L1 was saturated at 1.2 milligrams per kg dose or above of that of irzolizumab. When we incorporated this target pharmacodynamics data into mathematical models, what we have come out is that approximately 1.2 milligrams per kg dose of irzolizumab is sufficient to saturate tumor PD-L1 levels for 24 hours. Collectively, these data establish that target pharmacodynamics acquired at the tumor can be used for dose finding and dose optimization. In summary, what I have shown here is a pharmacodynamics-based approach to assess the activity of antibody therapeutics at the tumor and non-invasively. Importantly, this approach is agnostic to disease, location, and the type of the antibody used, and can be used for dose selection, optimization, and dose intensification of the antibodies. We hope that these kind of measurements could also enable quantitative relationships between dose, tumor and tissue exposure of the antibody therapeutic, and eventually relate that to response and toxicity. Furthermore, these measurements could be useful in drug development and evaluation in identifying antibody candidates that are engaging the target to the desired level in vivo prior to even getting them ready for clinical translation. Not only that, these kind of measurements could be applicable for antibody conjugates and other macromolecules, the studies which are ongoing. The work shown here is interdisciplinary, and by nature, very collaborative. We're very fortunate to have highly collaborative colleagues at Johns Hopkins and also across multiple institutions, and the support of the funding agencies, including NIH, DoD, and many private foundations. Also, thank you for your interest and your kind attention to our P41 webinar series.